Welcome everyone, and welcome to our virtual series. I'm Sandra Franco, and I am the Education and Outreach Coordinator for the Center for Integrated Cellular Analysis. And for those of you that aren't familiar with the center, it represents a collaboration from 10 different labs across New York City, interested in developing tools and methods focused on multimodal single-cell analysis. This series, this virtual series, represent where we aim to highlight papers and methods that stem from our center and that we think that might be of interest for the broader biological community. So before starting, I'd like to go over a couple of organizational details. So as always, we will have some time at the end of the talk for you to ask questions. You can either raise your hand and ask your question, or you can type the question in the chat box, and I will read it to our speaker. And with that, I'll introduce you to Tim Stuart. Tim is a postdoctoral fellow at the CETIGEA lab. He obtained his PhD in biochemistry from the University of Western Australia before joining the New York Genome Center as a postdoc in 2018. During his postdoc, Tim has led the development of new computational methods for the analysis and integration of single-cell data. I'll hand it over to you, Tim.
Thanks, Sandra. Okay, I'll share my screen. Great. Okay, thanks. So today, I'd like to talk about a package that we've developed in the lab called Signiac that enables the analysis of single-cell chromatin data. And we've designed this package particularly with a focus on enabling the analysis of multimodal data, where we might measure chromatin state alongside other modalities in the cell. So just to begin, I'd like to kind of highlight some of the analysis goals that we might have in analyzing a single-cell chromatin data set. So the first goal that we might have is really a basic goal that's kind of shared across many different single-cell assays. So this is to identify and annotate the different cell types or states that we have in the data set. And from there, we can go further and ask, what are the putative functional elements of the genome that are active in the different cell types that we observe. We can go a step further again and ask, within these DNA elements, what are the features of the DNA sequence that might be overrepresented and might correspond to transcription factor binding motifs, for example, that are active or bound by different transcription factors in those cells. We might also generate multiple single-cell chromatin data sets, and we want to analyze them together jointly in a way that allows us to make comparisons across these data sets and correct for differences, confounding variation differences due to technical variation between the data sets. So we need methods that enable us to integrate these data sets together. And finally, just a sort of general goal across the whole workflow would be to really enable the visualization of these data sets. And this is a little bit more challenging than, say, single-cell RNA-seq, because here our signal really covers the entire genome. And so we need to enable visualization methods that can pull information per single cell from any position in the genome rapidly and construct these visualizations. And we might also want to layer in additional information that we have from other assays measured in the cell, like gene expression, for example. And what's been really exciting to see in the field as well is the development of many different methods now that enable measuring not just chromatin state, but other modalities like gene expression, for example, within the same cell. And so what I'm showing here on this slide is just a variety of methods developed over the past few years that enable measuring DNA accessibility by scACT-seq alongside transcriptional information within the same cell. And what's interesting here is that the more recent methods like scACAT-seq and the recent 10x genomics commercial method are now approaching the level of sensitivity that we typically get when measuring each of these modalities in a single assay. But we're getting that same level of sensitivity now in a multimodal measurement. And this is really exciting because it can allow us to do a couple of things. So we can start to move beyond classifying cells based purely on their transcriptional state, So we can classify cells and analyze them based on multiple measurements that we've made from those cells. And we can also start to learn how these different modalities relate to each other. So in this example, we could try to learn how the chromatin state influences gene expression. So we can start to understand how different regulatory elements regulate the expression of genes and how that changes across different cell states. So we were really interested in these types of analysis in the lab, but there's a real need as well for computational methods that enable the analysis of multimodal data sets like this. So in the scATAC lab, we developed the toolkit scACT that was really originally designed for analyzing single-cell RNA-seq data. And the scACT object is really a collection of assays. So these assays are sort of the central data structure that we use. And it contains places to store a number of different things. So basically, we have a count matrix, a normalized version of the count matrix, a scaled and centered version, and we might have some other information as well, like feature metadata, which is could be information about the genes that we measured. And this is great for analyzing single-cell RNA-seq and other assays as well. But for single-cell chromatin data, there's a fundamental difference where the signal that we're interested in really spans the entire genome. And this kind of raises the need for a different data structure. So in Signiac, we've built on this standard assay in scACT, and we've developed an extended assay, the chromatin assay, that enables us to store some additional information that we need to run a full analysis of a chromatin dataset, like a single-cell ATAC-seq dataset, for example. So this includes things like the genomic ranges associated with each feature in our dataset. We can store the set of gene annotations that we're interested in. We can also store the path to on-disk data files associated with each of our experiments. So these are fragment files that are compressed and indexed and allow us to rapidly query different regions of the genome. And we can store other things as well, like DNA sequence motifs, for example. And so this data structure really enables us to run from beginning to end an analysis of these data sets. And the really important point here as well is that the way that we've designed this is we can store these chromatin assays alongside standard assays in the scACT object. And this is really conducive to doing a multimodal analysis using scACT and Signac together, because now we can store, say, our DNA accessibility data alongside gene expression data in the same object. Okay, so in this talk, I really want to focus on some of the functionality that we've built into the Signiac package. And I won't be getting too much into the biology, but I just want to highlight this data set that I'll be using sort of as an example throughout the talk. This is a data set that's publicly available from 10x Genomics, where they've profiled gene expression and DNA accessibility in the same cells. And shown here, we've processed the gene expression assay using scACT and the DNA accessibility assay using Signiac. And each of the cells here are the same in the two different plots. And we've annotated the cell types using the gene expression information. Okay, so how do we actually go about analyzing, say, a single-cell ATAC-seq dataset using Signiac? Typically, we would begin by computing a couple of different quality control metrics that we find informative when looking at ATAC-seq data, nucleosome signal and the TSS enrichment score. So the nucleosome signal metric here, we basically expect to see this sort of periodicity in fragment lengths collected from an ATAC-seq experiment. And this is because the TN5 enzyme, we know that it preferentially integrates into the genome in the linker DNA between nucleosomes. So there's an expected depletion of fragment lengths corresponding to the size of the length of DNA wrapped around a nucleosome. So we can capture this sort of periodicity in a metric, the nucleosome signal metric. We can also look at the TSS enrichment score. So this is computing the sort of ratio of TN5 integration events centered on a set of transcriptional start sites relative to counts flanking that region. And this is basically, it's a metric that originally defined by ENCODE. And it's useful because we typically assume that there's this accessibility at the TSS, especially relative to regions flanking that. So this serves as a sort of set of positive control regions where we would expect to see signal. So we can compute these metrics per cell. We can remove cells that might be lower quality based on these metrics. And basically we generate a counts matrix where we have cells and peaks. And here peaks could be potentially hundreds of thousands of peaks spanning the whole genome. And so the next step we want to do is reduce the dimensionality of that data set. And what we typically use in Signiac is a method called latent semantic indexing. So there are other methods that have been developed for dimension reduction, but this is one that we typically use. And this has a couple of steps. So one is we want to first correct for the differences in sequencing depth that different cells have. So we can do this in a kind of simple way by just computing the total number of counts per cell shown here in this local frequency vector, and then divide our count matrix by that. So now we have a sort of depth corrected, at least to some extent, count matrix. We then want to compute a set of feature weights because we know that not every peak is as informative for separating the different cell types in our data set. So if you can imagine a peak that's just ubiquitously open in every cell type, that's not really informative for separating between the different cell types. Whereas other peaks that might be open in only a subset of cells are more informative. So we can compute a set of weights, which is basically capturing that the fact that some peaks that are more ubiquitous should get a lower weight, and more rare peaks should have a higher weight. We then multiply those weights by the matrix here, and we get this TF-IDF matrix, which you can basically think of as a depth-corrected, feature-weighted version of the count matrix. And then we decompose that by SVD to get a low-dimensional representation of the data. Okay. So then we've done this linear dimension reduction step. We now have a matrix, which is cells by some number of LSI components, maybe 30 or 50. We can then further project those cells down into 2 dimensions for visualization. So we can use like UMAP, which is shown here, or TSNE, for example. And we can proceed with a whole range of different downstream analysis tasks. So we can find differential peaks between different cells. We can do DNA motif enrichment, TF footprinting. If we have multimodal data like we do here, we might link peaks to genes that they're correlated with. And we can run other tools developed by the community like ChromeVar or Cicero, or maybe Monocle for doing trajectory analysis. Okay, so I mentioned that there are several methods that have now been developed for dimension reduction for single-cell ATAC-seq. And using our multimodal data set, we wanted to start to evaluate how well these different methods perform. And particularly the LSI method that we've implemented in Signac is actually a slightly modified version of the typical LSI that we found performs slightly better when applied to single-cell ATAC-seq. So we wanted to also see how well our modified method does in comparison to some of these other methods. So the way we wanted to evaluate the performance here is we have a multimodal data set. We've annotated the cell types based on the gene expression information only. So this is independent of the DNA accessibility data that we're using for dimensionality reduction. And to evaluate the methods, we can basically ask how well do these different dimensionality reduction methods perform in reconstructing the neighborhood structure that we found using the RNA assay. And we did this for a range of downsampled versions of the ATAC-seq matrix to test also how these methods perform on lower sensitivity data. So shown here is the metric that we compute, which is just for each cell, how many of its k nearest neighbors belong to the same cell type as that cell. And you can see here the different methods as well that we looked at. So we have in blue, the LSI method that we're using in Signac. In orange is another very similar modification that was independently proposed, this log-TF method by Andrew Hill. We have scale, which is a deep learning autoencoder-based method, and scTopic, scACT-seq, which is based on the Jacquard similarity, and the original LSI method. And we can see on the full data set, they all do fairly well. Once you start to downsample the counts down to 20% of the original counts per cell, there becomes a substantial difference in the performance of these methods. The modified LSI methods do quite well. Scale also still does quite well. But methods like scACT-seq and the original LSI start to do poorly. And we can look at this kind of visually as well. So here we're showing a UMAP computed on the full data set using LSI dimension reduction, scale, and scACT-seq. And when we're looking at the full counts, we can see that they all do kind of similarly. But when we're downsampling to 20%, LSI and scale still do fairly well. scACT-seq really starts to struggle to separate the different cell types in the data set. It's important to note also that these methods have a substantial difference in their runtime. So LSI, because it uses the SVD, which is a really fast, highly optimized algorithm, performs in a couple of minutes on this data set with around 10,000 cells. Whereas methods like scTopic and scale, they require substantially longer. So they take more like hours to process the data. So we think that the LSI method that we've developed here is really performs well both in terms of accuracy and in speed. Okay, so now that we've reduced the dimensionality of our data set, we've annotated cell types. A really common goal is then to kind of dive in and explore the data. So we've spent a lot of time in Signiac building in these really rich visualization methods. So shown here is the CD8A locus, where on the top, we're looking at the DNA accessibility tracks that we've generated from the single cell data. So we can also dynamically regroup the cells into any kind of grouping that we want. So here in the CD8A locus, you can see the CD8 T cells have high accessibility. We can also layer in other measurements that we might have per cell. So here we have gene expression data. So we can stack on the expression of any different genes that we're interested in. So here we see CD8A gene is nicely correlated with the expression at the locus. The CD3D gene is a T cell marker and it's expressed in all the different T cells. So this is the single cell data that we have. We can also layer in other data that we might have for different regions of the genome. So shown here below is actually data that's being pulled from a bigwig hosted on UCSC, which is showing the PhyloP conservation scores per base. So we can see, for example, this region of the genome is highly conserved between different species. We can also look at different gene annotations. We can see the peaks that were identified in our data set. And down the bottom, we're also looking at the candidate cis-regulatory elements that were defined by ENCODE. So we can see how these line up with our peaks and the signal in our data set. And here we've colored the CCREs by the class that they belong to. So we can see that there's distal enhancers, proximal enhancers, promoters, and different things annotated. So this is really great for exploring the data and generating static plots like this. But we've also developed a fully interactive version of these plots. So we can create all the same plots, but in an interactive way that lets us kind of scroll through the genome, zoom in and out in different regions, and view the data that way, which we find is really a powerful way to just dive in and explore the data that we have. Okay, so another thing that we noticed early on when we started to look at many of these different data sets, single cell ATAC-seq data sets, is that the peak calling methods that we use can be really important and have an important impact on the accuracy of different downstream analysis tasks. So just as an example here, we have the CD8A locus again, and we have two sets of peak calls. In red is the peak calls generated by CellRanger, which is the 10x genomics software. And in gray are peak calls from max2, which we've built into Signiac the ability to run max2 from R and within your analysis. And what we see is there are many regions like this where we have several different peaks that are close together that are correctly identified as separate peaks using max2. But these regions are all sort of blended together using CellRanger. And so then it would be very difficult to identify these regions as being differentially accessible between the different cell types. So here we see these three peaks actually differentially accessible in these three different cell types. But in CellRanger, this is all blended into one region. So we really lose power in identifying this region as differential between the cell types here. So we always recommend, firstly, visualizing your peaks in these sort of genome browser views. And also nearly always we found that max2 gives a better set of peaks. You can also run max2 individually on each cell type in the data set and combine the peak calls into a unified representation. And that also helps in picking up peaks that are specific to rare cell populations. Okay, so we've seen that you can really run an end-to-end analysis using Signiac, but we also wanted to see how well these methods scale to really large data sets. So we sought out basically the largest single cell ATAC-seq data set that we could find. And this is a landmark data set generated by the Brain Initiative Cell Census Network Consortium. So this measured 734,000 nuclei in the mouse brain. And we were able to process this from end to end, so from raw data, and construct this UMAP using Signac. And the cells are colored here by the original cell type annotation provided by the authors. So we also wanted to see, how the key analysis steps in the Signac workflow scale with different numbers of cells, and also how they compare to other software that are available for analyzing single cell ATAC-seq data. So we looked at the ArchR package, which is another popular package for analyzing single cell ATAC-seq data. And we ran each of the key analysis steps in the workflow for Signac and ArchR for different downsamplings of the data from 50,000 cells up to the full data set. And you can notice that generally for most steps, the increase in runtime is somewhat linear. You can also see that using more cores generally gives you a big reduction in runtime. And you might notice as well that there is this object creation step, which for ArchR takes what seems like a very long time, whereas Signac is not zero, but is sort of on the order of a couple of minutes here, whereas for ArchR, it's more like hours. And that's because ArchR has to create these custom H5 files, which can take a long time. There is a benefit to doing that. So subsequent queries on the data, once you've created those custom files, are much faster. So you can see this when you look at using one core for ArchR versus Signac, is substantially faster. But what we also found is that once you start to use many cores, the bottleneck really becomes the reading from disk. And so you start to lose some of the advantages of creating these custom files up front once you pull in many different cores. And so in running an end-to-end analysis on this data set, using 8 cores, we found that Signac in this case slightly edges out ArchR in terms of runtime on the full data set. Okay, so far I've been talking about really analyzing a single data set, but really often, we want to make comparisons between data sets as well. So shown here, we have 3 different PBMC data sets. In red is the one that I've been talking about so far, this multi-ohm data set from 10x. In green, we have a second multi-ohm data set. This one was not fact sorted. And in blue, we have a PBMC data set. So there's no RNA information for this one. And if we take a sort of naive approach and just merge the data sets together, even if we make sure to quantify the same exact set of peaks in these 3 different data sets, we see that the cells separate by a combination of their data set of origin and their cell type. And so it's difficult here to identify what the equivalent cell states are across these different data sets. So it really raises the question, how can we jointly analyze multiple single cell ATAC-seq experiments in a way that removes the technical variation and allows us to focus on the biological variation in these data sets. So I mentioned earlier this LSI dimension reduction method. So this basically decomposes this normalized TF-IDF matrix into 3 different matrices. So previously we were kind of focusing on this matrix U here, which is the cells projected into a low dimensional space. And this is what we're typically interested in. But we also get these other 2 matrices, V and the singular values sigma. And these are really useful as well because we can actually use these matrices to project a new data set into the same low dimensional space. So here, for example, if we have a new TF-IDF matrix from a separate data set, we can project that data set into the same space as our original data set using these other matrices. So we have this LSI projection method that enables us to put different single cell ATAC-seq data sets into a shared space. And so what we've done now is extend the data integration methods that we've previously developed in the lab to use this LSI projection as a way of constructing a shared low dimensional space that enables us to identify anchors between data sets. So these are pairs of cells that we define as being equivalent cell states across the pair of data sets. And this allows us to do a couple of things downstream. So one, we can transfer information from a reference to a query data set. And secondly, we can integrate the data sets together to remove some of this confounding variation while preserving the biological variation. So we've now extended scRAT to include these methods for anchor finding. And now if we look at our example again with these 3 different data sets, this is our naive merging approach. But if we use LSI projection, anchor finding, we can integrate these data sets together. And now we see that the cells grouped by cell type, which is annotated here on the right, and these annotations are derived from the RNA assay in these data sets. And we see that the data sets or the cells group by their cell type and not the data set that they originated from. So now we've been able to remove some of this confounding variation and enable the common cell states to be identified, which would then allow us to compare across these data sets and make interesting comparisons. So another interesting thing that we might do is if we have an existing reference data set, maybe this is a very large, high quality single cell ATAC-seq data set, maybe it has some additional information that our queries don't have, we can actually take a new data set and map that to the query. So shown here is our PBMC multiome. We've annotated the cell types. We can then map our query single cell ATAC-seq data set onto that using LSI projection for finding anchors. And here we've mapped the cells onto the same existing UMAP as the reference. So we can kind of visualize the cells in that same low dimensional space. We can also transfer the cell type labels. So here we've been able to automatically annotate the different cell types that we have in the data set. And also because in our reference, we have gene expression information, whereas in our query, we only have the DNA accessibility assay. We can actually predict for each cell the expression of every gene in the genome and visualize that. So now we can look at different marker genes in the ATAC-seq data set. And this is the predicted expression patterns of different marker genes. So these methods really enable us to explore the query in the context of this existing reference data set and really enhances the interpretation of our query chromatin data. And interpreting these data sets is particularly challenging for chromatin data because we don't typically know as much about the non-coding, the function and the cell type specificity of these non-coding DNA elements. So enabling this reference mapping, particularly to multi-omic data, can really enhance the way we can interpret these data sets. Okay, and now just to summarize, we've developed Signiac, which is a R toolkit that extends scRAT and enables the end-to-end processing of multimodal single-cell data. We've also developed a modified dimension reduction method that really enables some fast and accurate dimension reduction for single-cell ATAC-seq. We've also developed really extensive interactive data visualization tools that let you browse the genome and see the signal per single cell for different regions of the genome. These methods also scale to very large data sets containing hundreds of thousands of cells. We can also do cell type specific peak calling on our chromatin data, and we found that the way that we do peak calling is really important in certain downstream analysis tasks. And finally, we've developed a new extension of the scRAT integration and reference mapping tools that enable single-cell chromatin datasets to be integrated and to build these references and also map query datasets onto these integrated references. And there's also a lot more that I haven't had time to talk about today, but we have a suite of analysis tools, for example, that enable the analysis of mitochondrial genome variants that can be informative for identifying different cell clones. That was in collaboration with Caleb LaRue. We also have tools to do things like DNA motif analysis and transcription factor footprinting, and also multimodal integration. So integrating ATAC-seq with RNA-seq datasets, for example, where you don't have a shared data modality like we saw in the case that I talked about here, but where there's no shared features, we can also enable a multimodal integration. Okay, so with that, I'd just like to thank our collaborators on the project and the members of the Satija lab, so particularly Rahul Satija, Avi Srivastava, and Shiosta Madad, as well as Caleb LaRue at Stanford. And if you want to find out more about the project, we have a manuscript that's on bioRxiv. We also have a lot of documentation and vignettes on the Signiac website. The package is also available on CRAN, so you should be able to install that fairly easily if you're interested. And we have the full code as well as discussion forums where we provide support on GitHub. So with that, yeah, I'll be happy to take questions. Thanks.
Thank you, Tim. So we have a lot of questions here in the chat. So I'll start with Anjali Chola. I think you can unmute yourself and ask your question.
Okay, hi, thanks. Thanks for this great tool and really helpful presentation. I have a few questions, so I'll just prioritize the ones that I'm interested in. So I know you've compared with ARC-R and scACT-seq, but from my understanding, they both use genomic bins versus just peaks for clustering. So have you done like a sort of analysis to see which performs better for clustering and what are the pros and cons for that?
Right, so in the comparisons I showed, we actually use the same regions for ARC-R and Signiac. So in that case, we were both looking at peaks. You can also use genomic bins. I haven't done a sort of comprehensive comparison of using peaks versus bins for, say, clustering the cells. My feeling is that they do fairly similarly, but the big difference is in some of these downstream analyses. So if you start to find, say, differentially accessible peaks or bins, you will find a difference in performance between using bins versus peaks, because when you're looking at peaks, you're really narrowing in on what the most relevant sequence of the genome is for that region, whereas peaks can include flanking sequence that might not be really relevant. And so when you start to do differential accessibility and motif enrichment, it's important to kind of really restrict the region that you're looking at to the region that's important and not include kind of flanking sequence that might decrease your power to find, for example, enriched motifs. But just in terms of clustering the data, yeah, they tend to do similarly, although I haven't done a comprehensive comparison.
Okay, yeah, thanks. And my next question was about the integration. I know at some point in the tutorial you had Harmony as an option to integrate. Do you think, do the reciprocal LSI integration and Harmony, they perform similarly? And also, would you, for integrating single cell ATAC, and would you recommend using single cell transform normalization before integration?
Right. So yeah, you can certainly use Harmony, and it's a great method. I think one of the things that we can do using the sort of LSI anchoring that you can't do with Harmony is the sort of reference mapping and data transfer stuff that I talked about. So that's something that we're particularly interested in, where you have big references that are high quality data sets that we've built and then map a query to it. Yeah, again, I haven't really done like a comprehensive comparison between Harmony and scACT for scACT-seq. My guess is that they both perform fairly well, and they're also easy to run. So you should certainly try running both and then see what kind of works well for your data set. And then, sorry, what was the second part of your question?
The SC transform.
Right, yeah, so yeah, we definitely don't recommend running SC transform on the scACT-seq data. So there's SC transform was really developed for count data that follows like a negative binomial distribution, which is not really true for scACT-seq. So for scACT-seq, we definitely recommend doing, if you want to go down the path of running LSI, you can do the TF-IDF transformation instead.
Okay, thank you. Thank you so much.
Next question from David Glass. How does PCA perform as the initial dimensionality reduction step?
Okay, so I think there are a couple of issues with running PCA on scACT-seq data. Firstly, you typically need to scale and center the data set for running PCA, and that transforms the data in a way that it's not sparse anymore. So there's a computational problem there where if you have, say, 200,000 peaks and 10,000 cells, you now have a huge non-sparse matrix that requires a huge amount of RAM to store. Whereas when you run the TF-IDF method, any values that start as 0 remain 0. So you don't have that issue. But in the end, the PCA and LSI are fairly similar, so they both use SVD. I haven't actually tried running PCA on an scACT-seq matrix, mainly because of the problem that it's hard to scale. And I think the other issue is that in PCA that there's no we don't have the same sort of feature weighting step that LSI tries to do. So we're just trying to explain the variance in the data. Whereas in LSI, we compute this inverse document frequency, which is a set of feature weights that would try to reduce the impact of peaks that are just uniform while upweighting the variable peaks, which I think works kind of better in the case of scACT-seq.
Thank you. Next question from Fulon Yu. What's the main difference between Signac, LSI, and the original LSI method? Maybe you can touch that very briefly.
Right, yeah. So it's basically it's just taking a log at a different stage when you're computing the TF-IDF matrix. So yeah, there's a full description in our preprint. But basically it's something like log IDF versus log TF times IDF, which just kind of happens to give a better distribution of the data in the end when I think mainly the difference is that the LSI method was originally developed for analyzing text. So if you have like a collection of books, and that data is much less sparse than ATAC-seq data. So in ATAC-seq, you have very sparse data, and the distribution is kind of different. And so it's just a small modification that we found gives better results. Yeah.
Bruce Aronow is wondering if a derived tract under user control would be helpful. For example, MAAT minus NK.
A derived track?
Maybe.
Yeah, on the peak, doing a peak difference analysis, and can you sort of prioritize a list of peaks that genome wide or region wide or something that could then kind of let you zero in on different cis-regulatory region peak structures between two related cells that you want to understand.
Right. You mean sort of visually when you're browsing the data?
Yeah, and maybe a table
right.
output of it too.
Right. Yeah. Yeah, I think it could be useful. One of the challenges is that when you're just looking at sort of raw counts, there's different numbers of cells in different populations, and how you subtract one value from the other, you'd have to correct for that, which, to be fair, we already do that when you're creating these coverage plots. Yeah, I think, potentially it's an interesting thing to look at, particularly visually, if you want to kind of quickly narrow in on things that are different between different groups. Sure, yeah.
Great, thanks.
Alex Rajewski, sorry. Oh, apologies. Alex Rajewski is asking, is it better to do the dimensionality reduction on the max2 peaks or the cell ranger peaks?
Yeah, so in terms of dimensionality reduction, kind of similar to the previous question, it doesn't tend to have a big impact on, say, clustering. But if you want to do differential accessibility and find peaks that are different between the different cell types, then it does have a big impact. Because if you're sort of merging together things that should be separate, you're really lowering the power that you have to kind of distinguish between accessibility levels between different groups of cells. So that's the case basically where we find it does become important. If you're just interested in clustering the cells, which typically you're interested in doing more than that, then yeah, you can use any set of peaks or bins, and the results don't tend to change very much.
For integrated, so RNA plus ATAC data set, is it always better to use the RNA data to annotate cell types rather than the gene activity data from the ATAC?
So yeah, in cases where you have like multimodal data and you have an actual RNA measurement, I think in that case, it's always better to use the actual RNA measurements. So yeah, we could, for example, look at gene activity for the multi-omic data set, which would be looking at how accessible the gene and its promoter is. But the goal of that is really to get an estimate of gene expression in the absence of actual gene expression measurements. Where we have gene expression measurements, then those are, that is the real gene expression for that gene in that cell. And so we should always use that. There is an interesting application where you can evaluate how well these multimodal integration methods work by creating that gene activity assay and then using it for integration, and then asking, how well do you annotate the cell types compared to how you would annotate them if you use the actual gene expression. But sort of beyond, using it in that context, yeah, we typically always use the real gene expression.
Will Signac work similarly for 10x genomics, single-cell multi-omic ATAC plus gene expression KIT dataset? Because it seems that the ones you presented are using KIT for single-cell RNA-seq and single-cell ATAC-seq separately.
Yeah, so actually the data set that I showed for most of the talk was the multi-omic. So having gene expression and accessibility in the same cell. So yeah, you can certainly use it for that. And that's, one of the main motivations that we had in designing the package in a way that allows you to use the package along with all the functionality in Seurat. So you can have a gene expression assay and a chromatin assay and one Seurat object, and then use the two packages to analyze them jointly.